activation of deoxyribonucleotides. Now, next step, exposure of DNA strands. Come on that. Next step is exposure of DNA strands. First, we activated our deoxyribonucleotides. Then what happens? At the position of ori, origin of replication, what happens? The two strands get separated. Okay, so the two strands get separated at the origin of replication site, and this is known as ori site. Okay, where it will happen? At the position of ori site. Okay, and here enzyme used helicase or unwindage. So by the help of enzyme, by the help of enzyme helicase or unwindage. Okay, so this you have to remember, that which enzyme used to separate the two strands. Okay, now previously it was joined like that. Now it become what? Single stranded, like that it is coming single stranded. So this single stranded having the, they want that again wind, so that here what happens, the separated strands are stabilized by SSBP, single stranded binding proteins. So what I told, the separated strands are stabilized by SSBP, means single stranded binding protein. Okay, so this doing what? Here single stranded binding protein comes and they don't again wind each other. Okay, so this separated strands are stabilized by what? Single stranded binding protein. Okay, now one more enzyme is there, that name is topoisomerase. Okay, so this doing what? The coiling having the capacity, so the coiling tension of these separated strands reduced by, that is reduced by enzyme topoisomerase. So reduced by enzyme topoisomerases. Very important, that a coiling tension of separated strand reduced by which enzyme? That is a topoisomerase. Now separated strand stabilized by which protein? SSBP, single stranded binding protein. Okay, Now, then what happens? This separation of two strands of DNA produced a Y-shaped structure. We seen a Y-shaped structure known as replication fork. What told? What I told? The separated strands of DNA forms Y-shaped structure known as, that is known as what? Replication fork. What I told? Y-shaped structure of these separated. We have we seen these separated structure forming like that. We seen now. So this Y-shaped structure that are forming, it is known as what? Replication fork. Okay. Now, if we are saying that prokaryotes, so in prokaryotes only DNA garage is there, that untwist DNA. Okay, what I told? In prokaryotes. Here in eukaryotes, what are the enzymes used? Here topoisomerase and helicase are used. Okay. So in prokaryotes, topoisomerases and helicases are replaced by, okay, so this topoisomerase and helicase that is used in eukaryotes, now it is replaced by DNA gyrase. Okay, so replaced by DNA gyrases. This also you have to remember that in prokaryotes what enzyme is used? That is used DNA gyrase. Now this DNA gyrase also untwist DNA. So DNA gyrase also untwists DNA strand. Okay, so this is doing what? DNA gyrase also untwist DNA strand. Okay, now the two separated DNA strand now act as template. What I told? The two DNA strands now function as template strand where replication or duplication is going on. Okay, so template strand having what? 3' to 5', and a new DNA formation occurs in the range of what? 5' to 3'. Okay, now next what steps is there? DNA polymerases involvement. So DNA polymerases involvement. Okay, next step is DNA polymerases involvement. So what happens? The DNA polymerase third Initially, we told it is the most important. So the DNA polymerase third is a common DNA synthesizing enzyme. What I told? DNA polymerase third is common DNA synthesizing enzyme. Okay, so DNA polymerase third is what? Common DNA synthesizing enzyme. Okay, now another DNA polymerase 2 and 1 is also used in replication. Okay, that also used in replication, but all of them function in which direction? So all these enzymes function in 5 prime to 3 prime direction. Okay, so all they are moving in the which form? 5 prime to 3 prime direction. Okay, now fourth step is RNA primer formation, or that is also known as initiation phase. So from that next step we will discuss in the next class. Okay, thank you.